Toxoplasma gondii. Toxoplasma is an obligate intracellular parasite, meaning it must stay inside a cell in order to survive. It usually stays in the cytoplasm of the cell. It was discovered by Nicole and Mansour in a desert rodent, the Gondii, at the Pasteur Institute at Tunis. Life cycle of Toxoplasma gondii. An infected rat contains brachyzoites of the parasite in its muscle tissues. There, they are enclosed by cyst walls and are called tissue cysts or zoetocysts. These tissue cysts get to the cat, the primary host, when they feed on an infected rat. And a primary host is a host where sexual reproduction of the parasite takes place. In the small intestine of the cat, the cyst walls of the tissue cysts would be dissolved by digestive enzymes, releasing brachyzoites. The brachyzoites would multiply rapidly by a special form of asexual division known as endodiogeny, a process where two daughter cells are formed within a mother cell which is then eaten by those two daughter cells. This rapidly dividing stage is now called tachyzoids, which is the same as trophozoites. Next, the tachyzoids will divide asexually again to form merozytes. At a point, these merozytes would start sexual division to produce gametes. Gametes fuse to form a zygote. The zygote develops into an oocyst, which is ingested in the cat feces. An oocyst is an egg-like structure which contains sporocysts, and those sporocysts contain spores or sporozoites. In addition to the brachyzoites in the muscles of rats and also birds, sporulated oocysts are another infective stage. Sporulation or maturation occurs in the environment and takes 1 to 5 days. Another cat could get toxoplasma by ingesting food contaminated with oocysts. The process would be the same, except that in place of tissue cysts, it would be oocysts containing sporozoites. Sporozoites multiply, forming tachyzoites, then merozytes, gametes, zygote, and oocyte. And note, this is all taking place in the small intestine, so it is called the enteroepithelial stage. When a grazing animal like goats feeds on vegetation contaminated with oocysts, the oocysts would enter the gut. Enzymes would free the sporozoites, which would multiply in many cells to form tachyzoites. As the immune system fights back, tachyzoid division would slow down and enter a resting stage, brachyzoites, which would encyst in tissues to form tissue cysts. As we can see here, there is no sexual growth, no gametes are formed. Therefore, other animals like goats are secondary hosts. Man can get it by eating undercooked meat from an infected animal. Tissue cysts would enter the body. Enzymes would free brachyzoites, which would multiply primarily in the lungs and liver, forming tachyzoites. As immunity kicks in, brachyzoites will begin to form and develop cyst walls in tissues like those of the brain and eyes. Man can also get it directly from cats by eating food contaminated with oocysts of the parasite. Oocysts will release sporozoites, multiply to form tachyzoites, and then brachyzoites. Mother-to-child transplacental transmission occurs in toxoplasmosis. This is basically how toxoplasma is transmitted. Pathology of Toxoplasma. Toxoplasma infections are mostly asymptomatic, except in immunosuppressed patients where the immune system has been compromised through illnesses like AIDS. Symptomatic cases could be acute, subacute, chronic, or congenital. Acute infections occur when the immunity of the host builds up very quickly to stop tachyzoid multiplication, hence the illness is short-lived. Subacute is when the immunity of the host develops more slowly than in acute, but it still develops, so it's a bit longer than acute. Chronic is where the immunity of the host takes a very long time to develop. Since it is immunity that drives brachyzoid formation, tissue cysts will begin to form at that very time. Reactivation of chronic infection is what commonly happens in immunosuppressed patients. Tissue cysts would rupture, releasing brachyzoites, which return to the multiplication phase, tachyzoites. That rupture is harmful and causes necrosis or death of cells. Diagnosis of Toxoplasma. The most widely used method is the cytoplasm modifying Sabin-Feldman dye test, a serological test. A serological test is one that diagnoses an infection by identifying antibodies produced in the blood serum in response to that infection. Another way is isolating the parasite from the blood, cerebrospinal fluid, or any other biopsy and injecting it into a rodent and then culturing it. Mice are ideal because they don't suffer from toxoplasmosis. A surefire diagnosis would then be a combo of the two, the Sabin-Feldman dye test with at least a four-fold rise in antibody titer and inoculation of the parasite in mice. Treatment drugs include pyrimethamine and sulfonamide. That's all. Thanks for watching.